my main positions that I push and like to push back on is insect protein. I think the insect protein is definitely the future. And then I talked about the bugs of war. That was recently, right? The bugs of war was about how insects have been used throughout history in combat situations. Insects also have traditionally had medicinal uses, and that's not changing with today's topic because, check this out, ants act as olfactory biodetectors of tumors in patient-derived xenograft mice. Abstract. Early detection of cancer is critical in medical science, as the sooner a cancer is diagnosed, the higher are the chances of recovery. Tumor cells are characterized by specific volatile organic compounds, VOCs, that can be used as cancer biomarkers. Through olfactory associative learning, animals can be trained to detect these VOCs. Insects such as ants have a refined sense of smell and can be easily and rapidly trained with olfactory conditioning. Using urine from patient-derived xenograft mice as stimulus, we demonstrate that individual ants can learn to discriminate the odor of healthy mice from that of tumor-bearing mice, and do so after only 3 conditioning trials. After training, they spend approximately 20% more time in the vicinity of the learned odor than beside the other stimulus. Chemical analysis confirmed the presence of the tumor changed the urine's odor, supporting the behavioral results. Our study demonstrates that ants reliably detect tumor cues in mice urine and have the potential to act as efficient and inexpensive cancer biodetectors. And it goes on to give us a nice little background run of what they're talking about here. Gives us some cancer statistics. Talks about how expensive it is to have these sorts of systems in place to early detect the presence. So let's see here. Methods, insects, and origins of colonies. Formica fusca is a relatively common ant species in the northern hemisphere. Colonies may contain several hundred individuals and are headed by one queen, monogamous, or several queens, polygynous. Three queen right colonies were collected in the forest of Emerneuville, France, gives the coordinates, and kept under laboratory conditions, gives the laboratory conditions, and in the laboratory of experimental and comparative ethology, and that's in the University of Sorbonne in Paris, Nord. Tested ants were foragers, ants that leave the nest in search for food, and were individually marked with dots of oil-based paint on the abdomen and or thorax 1 day prior to the conditioning. In all cases, each individual ant was used for only one conditioning and test experiment. Female immunodeficient mice, Swiss nude mice from Charles River Laboratories, were maintained under specific pathogen-free conditions of the laboratory of a preclinical investigation, again in Paris, France. the housing facilities were kept under the laboratory conditions, talks about light and temperature and all that stuff. The light dark cycle, triple negative breast cancer were established with consent of the patient. 12 mice were used in our study. Half were grafted with tumor at 9 weeks old into their scapular fat pad. The other half underwent the same surgical procedure, but without the graft of the tumor. The healthy sham groups, they went through the motions of performing the procedure on them, but did not actually insert the tumor, the graft from the tumor in there. And they all right. Urine of the mice was used as olfactory stimuli and was collected 7 weeks after the graft. Mice were placed individually in a cage without food and water for 3 hours. The floor of the cage was covered with filtered paper, which was collected at the end of the 3-hour soak with urine. Samples of 1 centimeter of urine imbibed filter paper was inserted individually in an Eppendorf tube and stored at negative 20 degrees Celsius, right? So here we have a pretty wild experiment going on, right? This is interesting stuff that we're able to do with, let's see, only growing the parts we want, thus making the entire process not only more efficient, but ethical as well, since we no longer need the entire cow to get a steak. Are y'all talking about lab-grown meat and stuff? Bioprint protein is the future. Oh, yeah. Got some fun stuff going on in the old side chat there. So yeah, basically they conditioned the ant to know which way the foraging, which way the forward, I don't know, call it foraging product, whatever they have them foraging for. Let's see. So I don't want to sit here and read the whole experiment to you guys, but what did they use as the bait? Because they would have made it so that following the trail of urine would lead to of the cancerous urine would lead to the bait. That's how they would train them, or condition them. Because hymenopterans are especially they have a very photographic memory, if you will. They remember everything. They can identify individuals. There's that funny TikTok of the guy saying that they had the wasp under the cup. Yeah, saying how it will remember your face and everything. That's not false. That's actually something that hymenopterans, like ants and wasps, are capable of. They have being eusocial. They have to be able to lead their group back to whatever it was and it give them this excellent visual mapping system. So yeah, they map your face when they look at you. Anyway, I'm not going to read the whole experiment, but you kind of see where it's going. How they talk about how they conditioned them to locate this, and hopefully this actually works, and we're able to get in league with the ants and fight cancer. So thank you for sticking with me today. Went a little bit over, not too bad. Usually I go about half an hour over, and I only went 12 minutes over today, so yay me. But there was a lot to talk about. Ants finding tumors is our palate cleanser over here on StrangeScience. I love our insect allies. Let's keep watching them.